So today we're diving into a technology that is completely changing how we see the human brain. It's this incredible breakthrough that lets us grow a brain in a dish, literally watching circuits form and function right there in a lab. And it might just hold the key to unlocking cures for some of the most complex disorders of the mind. Let's get into it. For more than 100 years, this has been the fundamental problem in neuroscience. The living, developing human brain, it's the ultimate black box. We can't take samples from it, we can't run experiments on it, and we definitely can't watch it get built from the ground up. So how did scientists finally find a way inside?
Well, we're going to get there in 5 steps. First, we'll set the stage with a really complex condition, the autism spectrum. Then, we'll dig into the great mystery that held back research for so long. After that, we'll look at the scientific alchemy that can actually turn skin cells into brain cells. From there, we'll see how those cells build working circuits in a dish. And finally, we'll connect it all to a future where we might be able to design targeted cures.
Okay, so to really get why this technology is such a game changer, we have to start with the kind of problem scientists are up against. And autism is the perfect example of just how big that challenge is. This analogy, it really hits the nail on the head. Think about it. Back in the 1800s, a fever could have been anything, a virus, a bacterial infection, you name it. The treatments were totally different, but they were all just lumped under one symptom. Autism is a lot like that. It's defined by observing behavior, not by a single biological test, which means it covers this huge spectrum of different underlying causes. And look, this isn't some rare condition. We're talking about a growing number of people. The number of diagnoses has risen to the point where almost 3% of the population is on the spectrum. That makes the search for its biological roots more urgent than ever. And, there are these really fascinating clues hiding right in the data. For instance, for every 1 female who's diagnosed, there are roughly 4 males. That is a huge difference. A biological skew that strong tells us that deep genetic and developmental factors are definitely at play. But the question has always been, what are they? Our search for the cause has been, it's been a journey. We went from these totally outdated psychological theories in the 50s to groundbreaking twin studies in the 70s that proved there was a strong genetic link. And now we're in the modern era where we've identified hundreds of genes associated with different forms of autism. The evidence is just overwhelming. It points to biology, but that brought us right back to that same old problem. So even when you know which genes might be involved, how in the world do you study what they're doing to the one organ you absolutely cannot get to? This quote just says it all, doesn't it? The frustration scientists have felt for decades. I mean, you can't perform a biopsy on a living brain without causing serious, permanent harm. And that one simple fact has been the single biggest barrier to making real progress in treating psychiatric and neurological disorders. Just think about it for a second. We've made these huge leaps in treating something like leukemia. Why? Well, because we can easily get blood samples to study in the lab. But the brain? It's locked away inside the skull, especially during those critical periods when it's first developing. That inaccessibility is exactly why understanding the brain has lagged so far behind other areas of medicine. But then, something happened. A discovery came along that was so profound, it was like someone finally handed scientists a master key. A key that could finally unlock the brain. It was a discovery that would change absolutely everything. The whole idea, it sounds like something out of an old alchemy text, right? For centuries, we thought biological development was a one-way street. A skin cell was a skin cell, and that's all it could ever be. It could never go back to being a powerful, primitive stem cell. But what if you could force it to? What if you could literally rewind its developmental clock? And this is what they came up with. Induced pluripotent stem cells, or iPSCs for short. Now, this is a really important point. These are not embryonic stem cells. These are adult cells taken from a person's skin or blood that have been scientifically reprogrammed back to a state where they can become anything: a heart cell, a liver cell, or, most importantly for us, a living human brain cell. And the process itself is it's incredible. It all starts with a simple skin sample from a patient. Then, scientists add 4 key genetic factors. They're called the Yamanaka factors. And these act like a reset button. They tell that specialized skin cell to forget what it is and go all the way back to being a versatile stem cell. From there, scientists use a specific cocktail of molecules to guide that stem cell to become a neuron.
all while carrying the unique genetic blueprint of the person it came from.
Okay, so making a single neuron is one thing, but the brain isn't just a pile of cells, right? It's all about the connections, the circuits. So the next logical question was, could these lab-grown cells actually get together and build those circuits?
And this is where things get really wild. It turns out, when you let these stem cells float around in a 3D culture, they don't just form a random blob. They self-organize. They start to form these tiny structures that actually resemble parts of a developing brain called organoids. And what's even crazier is that they seem to follow an internal schedule, an intrinsic clock, maturing at pretty much the same pace as a real brain developing in the womb. So from there, scientists took it to the next level with something they call assembloids. The idea is actually pretty simple, but it's incredibly powerful. If you can grow an organoid that's like the cortex and another one that's like the spinal cord, what happens if you stick them together? Well, all of a sudden, you can build and watch the specific circuits that control everything we do. A motor circuit is the perfect way to see this in action. Scientists grow a cortex organoid, and they grow a spinal cord organoid. They fuse them together in a dish, along with some human muscle cells. The neurons from the cortex part reach out and connect to the spinal cord neurons, which then connect to the muscle. And the results is mind-blowing. When they stimulate the cortex part of this little structure, the muscle actually contracts. They have rebuilt a functional human pathway, completely outside of a body. And this is what brings us all the way back to where we started. Because this isn't just some cool science experiment. This technology directly tackles that huge, decades-old challenge of finding cures for devastating brain disorders. So now, instead of just guessing what a genetic mutation does to the brain, scientists can take a skin cell from a patient, build a working model of their brain circuit in a dish, and essentially watch the disease play out. For the very first time, we can see the biological problem right there in action. Timothy syndrome, a condition linked to a severe form of autism, is a perfect case study for this. When scientists grew neurons from patients, they saw exactly what was going wrong. See, in healthy neurons, a specific calcium channel opens and closes really fast to let the right amount in. But in the patient's neurons, that same channel gets stuck open, flooding the cell with way too much calcium and messing up its function. And for the first time, a complex psychiatric disorder was traced back to a clear, physical, mechanical flaw. You can see how this just opens up a whole new era for medicine. We can now pinpoint the exact biological problem. We can test potential drugs directly on a patient's own living circuits to see what actually works. This allows us to develop truly personalized, targeted therapies. And ultimately, this approach promises to demystify psychiatric disease, turning these abstract conditions into concrete biological problems that we can actually solve. I mean, this technology, it's one of the biggest leaps forward we've ever made in understanding that 3-pound universe inside our heads. And it really leaves us with this huge question for the future. If we're learning how to rebuild the brain's most fundamental circuits, what are the real limits to what we can one day repair?